Ames test aims to test whether avoiding a certain chemical might be best.
I say might be best because what the Ames test does is it tests to see if something is a mutagen. So if it can cause mutations to DNA. It cannot tell you if something is a carcinogen, that is, it can cause cancer. Bacteria can't get cancer, and so really what you're just measuring is mutations. If mutations happen in your body, and mutations happen in your body all the time, these mutations normally get fixed. Additionally, your body might even like break down a chemical before it can even hurt you. And so this test, it can tell you if something maybe has the potential to be a carcinogen, because if something causes a mutation in DNA, and then that mutation gives cells like a growth advantage, then those cells can multiply, and this is how you can get cancer. But remember, mutations are happening all the time, and something that causes mutations in bacteria is not necessarily going to cause cancer. So the Ames test is just like an initial test. Additionally, sometimes things that are carcinogens, so that can cause cancer, don't give a positive Ames test. And as addition to the like vice versa effect. So, but the Ames test is like a quick test that people can do, and it's often used as part of like regulatory procedures if people are trying to apply for permission to use some chemical or something like that. So it's an important test, and this is how it works. Basically, it takes these bacteria and that cannot use histidine, that can't make histidine, this amino acid histidine. We'll go way into more details, but so one of these protein letters, it can't make this important building block that it needs. And then you grow it in media, so bacteria food that doesn't have histidine, and then you measure the ability of mutations to form that will cause revertant mutations. So that will make these cells, it'll fix the gene that they had a defect in that was making them so they couldn't make histidine. Now they can make histidine, and since they can make histidine, they can grow on the plate, and then you can measure the number of colonies. And you can add like rat liver extract in order to test if something is like would get metabolized by the liver to make it mutagenic or non-mutagenic. And we'll get more into this in a minute. But here's the basic idea. So the reason why mutations can cause problems is that mutations have the instructions for making proteins as well as like functional RNAs and stuff I'm not going to get into, but it's easiest to think in terms of the proteins. And so you can think that if you made a mutation in one of these, the genes for a protein, then you can make a mutated protein, and then your body can't use that protein to do the stuff it needs. And so we often think about protein, if you were to look on like a label of food, it would just seem like this like protein is just like some generic-y thing. But really proteins are like the important workers in your cells. And so they're important proteins. I'm doing all sorts of important things. And if you cause a mutation in one of these proteins, then it might not be able to do those important things that it needs to do. Everything from putting together nucleic acids, so like DNA and RNA, to building other proteins, to holding cells together. Proteins do all sorts of really cool stuff. And so if you hurt those proteins, you could hurt the cell. I should also mention that, as I talked about in my post yesterday, if these mutations are happening in like a germ cell, so like a sperm or an egg, then those can get passed on and inherited. If they're happening in one of your somatic cells, so one of the cells just throughout your body, then it'll only get passed down to the cells made from that original cell. Also, as I mentioned yesterday, mutations are happening all the time, and most of the time they're no big deal. Most of the time they're fixed. But what about in bacteria? So bacteria, they're growing really, really fast. And this makes it so that if a mutation occurs that gives them a growth advantage, you're going to be able to see this really fast, especially if you are selecting for it so that they can only grow if they have that mutation. So because of the fast replication time, you have more possibilities for the mutations to be introduced because you're selecting for the cells so they can only grow if they get a mutation that makes them be able to grow in histidine. Then you're going to be able to see these mutations. And so this makes bacteria really good for doing these experiments. And we'll get more into the details about the types of bacteria that are used. They're typically some sort of like salmonella that is made harmless to you, and also deficient in repair and that sort of thing. So you take these cells. So these cells normally can make histidine. So because they're intact DNA instructions for making it. And so when something is like this, when something can make something it needs, we call it autotrophic. So this would be a histidine autotroph or whatever. So it can make histidine itself. And so it's going to be able to grow in a plate that even if the food doesn't have histidine, it doesn't need it from its food, it can make it itself. And so there's this like original mutation, and this mutation is going to be in a gene that's required for making histidine. And so now if you were to grow them on histidine, they wouldn't be able to grow. And so we call these oxytrophic for histidine. So you take these oxytrophic cells and you grow them on a plate without histidine. So the only way they're going to be able to grow is if they get some sort of mutation that fixes that original mutation. So this revertant mutation. So mutations are always going to happen at some baseline rate. But if you add something, a chemical that is a mutagen, then what's going to happen is that those mutations are going to be more frequent. Often these mutations are generally random. And so there's going to be mutations that are just going to like kill the cells and that sort of thing. There's going to be all sorts of off-target mutations. But if the mutation rate is random, then the rate at which these mutations randomly happen in this histidine needing gene, the gene you need for making histidine, then they'll also be more likely to occur there as well. And therefore, if you count the number of colonies, you can then compare the number of colonies that you get for under different conditions with different chemicals. And the more you get, the more potentially mutagenic the thing is. So how does this work in practice? You need at least like 4 plates. We'll talk later about how there's actually different strains of bacteria that you can use, and so you're going to actually want more. One is going to be your just like total control, your background control, and this is just going to be like natural mutation rate. So mutations are happening all the time. So bacteria, they're replicating really fast. Machinery is not perfect, and so they sometimes can get mutations, especially in these cells, which typically use strains that have problems with their fixing machinery. So basically, you want to know how many mutations to expect, if just like in the general growth of bacteria. So expect this many colonies, even if the chemical is like totally safe. Next, you want to see, okay, is it directly mutagenic? So is it mutagenic without having anything else, like any modifications to it? If it is, then if you add it, you're going to get a lot more colonies. So these colonies are these just like individual bacteria that take home on the plate, and then they grow on top of each other and they get this booby clumpy colony. So the more colonies you have, the more mutagenic something is by this test. Okay, but what happens if you actually put something in like a body? So bacteria don't have all the sorts of, don't have livers. And why I mention livers is because our livers are like a key place where metabolism occurs. And so metabolism is like the making and breaking and changing of molecules. And so what happens is that your livers modify a lot. Your liver modifies like a lot of different things in a lot of different ways. This can be like oxidation, so adding oxygen, so and all sorts of different things. Sometimes this makes things more soluble so that you can more easily excrete them. Sometimes they convert them into other forms that are less toxic. So your body's like constantly ingesting like weird stuff, and your liver's doing other, making it into less harmful stuff usually. You can also sometimes make it into more harmful stuff. It's not like, because the liver's, I mean, it faces all these different compounds, and it's not like it knows what all these different compounds are, but it has these basic enzymes, so these reaction speeder upper helpers that have these basic modifications that they can make to molecules. And so if the molecules, sometimes it can make these modifications to molecules without even like knowing what the molecule itself is. And so it's not like it knows that it's making something safer or that it's making something not safer. It's just making these modifications because, oh, the molecule matches my active site. So like the part of me that binds to substrates, so it binds to things and modifies them. And so what can happen is that the liver can either make something less harmful, or it can actually make something more harmful, or it can just like change it to another form. But sometimes what can happen is that a chemical that you add isn't directly mutagenic, so it can't cause mutations as it is, but something that the liver does to it in the metabolism of it makes it so that it is mutagenic. And so this would be like pro-mutagenic, and then your liver would make it mutagenic. So how do you test for this? So bacteria don't have livers, and so what you can do is you can supplement it with liver extract. So typically this is taken from like some sort of rodent liver, so like a rat liver extract. And what you would do is you would add this extract to the chemical and allow the chemical to get modified to see if the modified form would then cause mutations. And then you're going to want to compare this to, basically now you need a different control. So this would be the same, kind of like your background control here, except here you also have the liver extract, just to make sure that the liver extract itself isn't changing the background mutation rate. So you have a direct comparison. And then if you see more here than here, then this would tell you that it was pro-mutagenic. So that the liver, the metabolism of it was making it a mutagen. Okay, speaking of mutagens, there are different types of mutations. So we talked a lot about this in yesterday's post, but so you can have mutations that are basically swapping letters. You can have mutations that are inserting or deleting letters, and you can have mutations that are not making any changes really, at least at the protein level. So there's a bunch of different types of mutations, and different mutagens are more likely to cause different types of mutations. And so therefore you often use different bacterial strains in order to detect different types of mutation, like different types of mutations, since different mutagens might have a propensity for introducing, say, a point mutation, so just like a simple letter swap, or like a base pair substitution is another word for that, or like a frame shift. And so what happens is that, so this technique was originally developed in 1973 by the UC Berkeley scientist Bruce Ames. And since then, there's been a lot of modifications, including the use of a bunch of different bacterial strains that can detect different things. So often these are used in like Salmonella strains. We'll get more into the details about these strains in a minute, but you can see that there's these different strains which have these different mutations in that gene for making histidine. And these different mutations are then going to cause them to be able, they require different types of mutations in order to like fix them. So some of them just require like a single letter to be changed back, whereas some of them require a frame shift mutation. So what else is special about these cells? So I took this information from this really great article that is open access, and I'll share a link to. But basically, a lot of tests, let me just point out some important stuff. So these most important common tester strains. So they have different things that make them more likely to take in chemicals and to be able to less likely to fix the mutations, as well as mutations that are going to make them less harmful to you when you're working with them. So this UVRB mutation eliminates accurate DNA repair and makes the cells biotin dependent. This RFA mutation affects the bacterial cell wall, resulting in a defective LPS layer that provides more permeability to bulky chemicals. So it makes it easier for things to get in because it has mutations in this lipopolysaccharide cell wall. So this makes it more easy for those chemicals to get in, which is another reason why this isn't like a direct measure of what might happen in your body, because in the chemicals, they might have a different like trouble or easier time getting into your cells and getting into bacteria cells. So they have this PKM101 plasmid that provides ampicillin resistance. And so this allows you to select for them in other than like with the histidine. Basically, you want to have some way to make sure that you're only allowing these cells to grow and not other bacteria. And so we talked a lot about antibiotic selection in the past, where basically your cells, they now have this plasmid that has the antibiotic resistance gene. So if you grow them in the presence of the antibiotic, they'll still be able to grow, whereas things that don't have it won't grow. And it also helps with mutagenesis with error-prone DNA repair pathway. Okay, one of them has like tetracycline resistance. So tetracycline is another antibiotic. Blah, blah, blah. Yeah, so there's basically some other strains that you can use to do look for different types of mutagens. Okay, so that's the basics of the Ames test. So you take bacteria that are oxotrophic, so they cannot make histidine, and then you add a chemical and see if that chemical causes a reversion mutation that makes them autotrophic again, so that they can make histidine. If they can make histidine, then they can grow, even if you don't feed them histidine. And this allows you to see if something is potentially a mutagen, but not whether something is a carcinogen. And to do that, it typically uses like animal testing and that sort of thing, which I'm not going to get into. But what you can do to get one step closer is to add rat liver extract or some other liver extract in order to see if something is pro-mutagenic. So would the liver potentially make it harmful if when it like metabolizes it? Okay, so I hope that helps you understand this test, some of its limitations, and how it can sometimes be used as just like a quick test for potential mutagenicity. Okay, so hope that helped.